Welcome back, everyone. So we are on part 2 of topic 3, the building blocks of life. So in this topic, we're going to be looking at what is passive transport. So we're going to see how, since we already know how the cell membrane works, we're going to take a look at how things enter and leave the cell membrane. So, it's selectively permeable, how certain things can go in and out. And so we'll learn about that through passive transport. So by the end of this video, you should be able to describe the process of osmosis and diffusion, as well as define the following: isotonic, hypotonic, and hypertonic. You should also be able to define what turgor pressure is and plasmolysis, which has to do with cell walls. And then describe the factors that affect how molecules move from one location to another location. And then, since we're talking about cell walls, you should be able to identify the function of the cell wall. So first off, let's start out with, well, what is passive transport? So in the previous video, we talked about the plasma membrane in detail. So remember how the plasma membrane is a phospholipid bilayer. And because it's a phospholipid bilayer, it has that hydrophilic head and hydrophobic tails. And the way that it's situated is that it's selectively permeable. So it allows certain things to come in and certain things to not. Okay, so it's very selective of what can pass through. So there are certain substances that can pass through the membrane via something called passive transport. So passive transport, passive means no energy is required. So it just uses basically just, regular potential energy, no cellular energy is involved. Okay, so doesn't have anything to do with ATP, which is cellular energy. So this is just no energy, just something called diffusion. So what is diffusion? So this is when we have substances just have the tendency to move from areas of higher concentration to areas of lower concentration. So for example, think about if you have a air freshener spray, and when you spray that air freshener, at first it's like highly concentrated where you spray it, but then it diffuses out and spreads throughout the room, right? So it goes from an area of higher concentration, the area where you just sprayed, and to an area of lower concentration to kind of spread out. And so that's what diffusion is. So the odor particles are moving from higher concentration to lower concentration. And so they follow this concentration gradient. So a concentration gradient is just a physical space with a different concentration of a single substance. So substances have that tendency to move from a higher concentration to a lower concentration. So let's talk about selectively permeable, or mainly what is selectively permeable for our plasma membrane. Now we already know that the plasma membrane has the hydrophilic heads and the hydrophobic tails. And because of that, it allows certain substances to pass through. So here are a list of some of the substances that can pass through pretty easily. So we have lipid soluble materials. Okay, so because of the hydrophobic tails, things that are also hydrophobic can slip through that semi-permeable membrane. So things like vitamin A, vitamin D, vitamin E, vitamin K, those are all like fat soluble. If you've ever heard of, those vitamins, they're fat soluble, and so they can just easily kind of move through the plasma membrane. Also molecules that are very, very small with no charge. Okay, so we're talking really, really small. So for example, oxygen gas. So O2, that's basically just two oxygen atoms stuck together with a double bond. Also carbon dioxide. So just, this is one carbon with two oxygen atoms. So very, very, very small. So they can easily slip through. And if you think about it, those are the gas particles that leave and enter our cells. So when we breathe in, we're bringing in fresh oxygen to our cells, and then we're breathing out carbon dioxide. So those can easily pass through our cell membranes. Water, H2O, can easily pass through our cell membranes. Okay? And then some of our smaller ions. So remember, ions are those charged molecules. So they can easily slip through as well, or charged atoms. They can easily slip through as well. Okay. So let's talk a little bit about diffusion. So we already know that diffusion is a type of passive transport, in that it allows substances to move from an area of higher concentration to an area of lower concentration. But that doesn't mean that it's just basically when you spray that air freshener that it's just leaving that, the area completely and moving away forever. It's basically just diffusing until the entire area is equally concentrated, okay? So you can kind of see where we're equally spacing everything out so that everything has an equal amount. So we're moving everything from higher concentration to lower until there's an equal amount on both sides. Okay, so you can kind of see in this picture, we started out with an area of high concentration of this particle to an area of low concentration. And then slowly over time, we can see that the particles kind of diffuse from that higher concentration to lower concentration until it reached equilibrium. So in equilibrium, we still have movement of these particles moving across, but they're moving at equal rates. So some of them are moving this direction, some of them are moving that direction. They're just moving at an equal rate. Okay, so there's still diffusion going on, it's just at an equal rate. It's not just going in one direction right now, it's going in both directions, but it's a net zero. Okay, and because this is passive transport, no cellular energy is being exuded right now. So this is just passive transport. Now there's different things that can affect the rate of diffusion or how fast it goes. So some of it is the extent of the concentration gradient. So think about how much there is. Okay, so if we have quite a bit concentration of the molecules on one side of the membrane, then it's going to increase the rate of diffusion. Okay, so if there's just a little bit on one side, it might take a longer time for it to diffuse across. So the extent of the concentration gradient can affect the rate of diffusion. Another thing that can affect the rate of diffusion is the mass of the molecule diffusing. So, the bigger the molecule, the fatter the molecule, the heavier the molecule, the longer it can take to cross the membrane. So, think of, a hippo. It's going to take a hippo a while to cross the membrane compared to a little mouse. So it's got a lot more mass to get across. So it might take them a longer time to get across. So the mass of the molecule diffusing does have an effect on the rate of the diffusion. Temperature can affect the rate of diffusion. So if we increase the temperature, these molecules are going to start speeding up, and because they're speeding up, they're crashing into each other more, and they're going to make their way across the membrane at a faster rate. If we slow the temperature down or decrease the temperature, that slows those molecules down and it makes it take longer for them to diffuse across. Okay, so that will decrease the temperature. And then the solvent density. So basically, if we have a more viscous material solvent, then it's going to be harder for the molecules to make it across. If the liquid is more thick and viscous and highly viscous. If the liquid, the solvent is thinner, less viscous, it's going to be easier for the molecules to move across. Okay, so solvent density does have an effect on the rate of diffusion as well. So imagine these, molecules are moving through honey versus liquid water. Okay, so it's going to be a lot easier for them to move through liquid water than it is through honey. I'm going to watch a video. This is a video that's linked in your Adigia textbook, but I thought it's a good video to watch that just kind of explains this whole concentration gradient and passive transport concept that we've been kind of discussing so far.
Diffusion can also occur through cell membranes. However, cell membranes are selectively permeable. This means that some substances can diffuse cell membrane while others cannot. The flow of ions or molecules through a cell membrane as a result of diffusion is referred to as passive transport. It is referred to as passive transport as it requires no energy from the cell in order to carry a substance across the membrane. In the process of passive transport, the membrane acts as a filter. Phospholipids and protein channels control which molecules can cross the membrane and may influence the rate and timing of the movement of ions and molecules across the membrane. But they do not influence the direction of movement. Substances continue to move only from areas of high concentration to areas of low concentration. Let's now look at three types of passive transport that occur as a result of diffusion: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is the process by which gases, such as oxygen and carbon dioxide, dissolve in water, and phospholipid-soluble molecules, such as ethyl alcohol and vitamin A, diffuse across the phospholipid bilayer. This is referred to as simple diffusion because these substances can cross anywhere along the phospholipid bilayer, while most other types of diffusion, such as facilitated diffusion, which we will look at in a moment, requires a substance to cross the membrane through a protein channel or with the help of a carrier protein. Generally, the rate of simple diffusion of a substance across a membrane is a function of the relative concentration of the substance on either side of the membrane, the size of the substance's molecules, and on that substance's lipid solubility. In the process of facilitated diffusion, either channel proteins or carrier proteins facilitate the crossing of a cellular membrane by water-soluble substances such as sodium, potassium, and calcium ions, amino acids, and monosaccharides, which cannot typically diffuse through the phospholipid bilayer. As we saw earlier, each type of channel protein is typically lined with specific amino acids and has a unique diameter in order to allow only certain ions or other substances to pass. Channel proteins also often have gates which open and close in response to electrical or chemical signals. Carrier proteins, as we saw earlier, attach to specific molecules in the cytoplasm or extracellular fluid. The attachment triggers changes in the shape of the carrier protein that allows the molecule to pass through the protein and thus cross the plasma membrane. Some carrier proteins don't use cellular energy and only allow ions and molecules through the membrane that would normally pass through as a result of diffusion. However, as we will see later, there are carrier proteins that use cellular energy to carry substances against the flow created by diffusion.
Okay, so that video gave us, a good kind of review of passive transport, and also, defined it as simple diffusion, but also gave us a glimpse at facilitated transport. So facilitated transport or facilitated diffusion is also a form of passive transport. but in this case, or excuse me, of diffusion. But in this case, it requires one of those protein channels. So you can see how, along with our phospholipid bilayer, we have these protein channels embedded in the cell membrane. And they allow molecules that cannot just simply diffuse across the plasma membrane to go through as well. Okay, so they're still going from high concentration to low concentration. But they just have to, have some sort of a transmembrane protein to allow them to do that. So these are molecules that are either insoluble, they can't cross easily, maybe they're too big to cross, so they need one of these channels to open up. Okay, so it could be a carrier protein, it could be one of these transmembrane proteins that just opens up and allows them to cross through. And just like the regular old passive transport or passive diffusion, the materials are still moving from higher to lower concentration, and oops, that's misspelled. No energy expenditure is used. Okay, so this is just simple passive transport. Okay. And then the last type of passive transport we're talking about is osmosis. So osmosis is also diffusion, but in this case we're specifying the diffusion of water through a semi-permeable membrane. So just like any other substance, water in this case moving from a higher concentration to a lower concentration. Okay, so this kind of depends on the solute that's dissolved within the water, and the water being the solvent. So if we take a look at this beaker here, let's say that this red thing right here is the semi-permeable membrane. So you can see that the semi-permeable membrane is permeable to the water, but is not permeable to whatever these little purple balls are. Okay, so the purple balls cannot move, but the water can. So we can see on this side of the beaker, we have, quite a bit of water. The ratio of water to purple balls is, different than on this side. So if we kind of just create like a little ratio here, let's say that I have, I don't know, maybe 10% of this side is purple balls. Okay, the little, balls, B for balls. And then that means that 90% is water. I'll do W for water. Okay, so on this side, it's a lot of these, little purple balls. So I'd say 90%, and the rest is water. So that means that in this case, the water, it's less concentrated on this side, on this side compared to this side, is more concentrated on this side. 90% is water over here, only 10% of water over here. So water is going to go from higher concentration to lower concentration. So the water is going to want to even out. It's going to want to spread out to this side. So water is going to flow to this side of the beaker. And in doing so, you can see what happens. Now it's more evenly spread out. And so that's what osmosis is. It's the diffusion of water moving from higher concentration to lower concentration. So when we're talking about tonicity, tonicity is looking at the amount of solute in the solution. So this kind of relates to osmosis, because we're comparing the total amount of solutes dissolved in a specific amount of a solution. And so we can measure the tonicity based on that kind of ratio. And so if there is a lower concentration of solutes, outside of the cell or outside of, whatever we're looking at compared to inside the cell, we call that a hypotonic solution. So think hypo as low concentration of solutes outside of the cell. And so what happens is because of the lower concentration of solutes, that means we have a higher concentration of water. Okay, so if we have lower concentration of solutes outside of the cell, we have higher concentration of, so remember hypo means low solute. Low solute. So if I have low solute, that means that, so outside of the cell, we have low solute, so, and high water. Low solute, high water. Compared to inside. Low solute, high water. And since we have a higher concentration of water outside of the cell compared to inside, the water is going to want to move into the cell. So what happens is the cell is going to expand and it's going to gain water. And with our human cells, what can eventually happen is that they can gain so much water that they burst, or the technical term is that they lyse. Okay, so that's what happens in a hypotonic solution. Now the opposite is a hypertonic solution. So think hyper as high solute. Okay, so in a high solute, we have high solute. It's hard to write with the mouse. Low water outside of the cell. So if I have low water outside of the cell compared to inside, then the water is going to leave the cell because there's higher concentration inside than outside, and water moves from high concentration to low concentration. And so water is going to leave the cell. And as it leaves the cell, look what happens. It's going to shrivel up, or technically, the technical term is crenate. And so it loses water and it kind of shrivels up. Okay, so that's what ends up happening to our cells when it loses water. Now, we want it to be isotonic. So an isotonic solution means equal. So think iso, equal. Equal. I'll just do the equal sign. It's easier. So equal means equal concentration of solutes inside as outside. So if we have equal concentration of solutes, that means we have equal amounts of water. So water will be moving inward as well as outward at an equal rate. Okay, so it's not just, it's still moving. It's not just not moving at all. It's moving, but it's moving at an equal rate. Okay, and so this is where our cells want to be. They want to be in an isotonic solution. Okay, so we have the equal amount of water going in and out. Okay, so hypotonic, hypo, think low solute, high water. Water enters the cell, causing it to burst. Hyper, high solute, low water. Water leaves the cell, cell shrivels up. Iso, equal amount of solute, equal amount of water. Water equally goes in and out. Cell is happy. Okay, so those are the different tonicities or amount of solute in a water. Okay. So when we're talking about animal cells, since they don't have a cell wall, the cells can gain so much water that they burst, or they can crenate and shrivel up. Plants are a little different because plants can be protected due to their cell walls. So they can actually prevent themselves from bursting. From lysis. So what happens when plants are in a hypotonic solution? So let's take a look over here. Hypotonic, remember hypo means low solute, high water. So water is entering the cell. But look at what happens. The cell, here's the cell wall in black, and then you can see the blue line, or is that a green line? I can't tell. The blue or green line right here is the cell membrane. And look at the cell membrane. It's pushed right up against the cell wall at this point. And what happens is that we have something called turgor pressure. So the hypotonic solution ends up, as the water's coming in, it's putting this turgor pressure on the plant cell wall. And that's preventing the cell wall basically stops more water from entering. There's no more water that can enter because there's no more room. And so that prevents the cell membrane from bursting. Okay, because there's simply, the cell wall prevents more water from being absorbed. So the cell wall, in a sense, is protecting the cell from bursting. Okay, due to that turgor pressure on the plant cell wall. Okay, now if we look at a hypertonic solution, remember hyper, high solute, low water. So water is leaving the cell. Look at what happens to the cell membrane. So the cell membrane is shrinking away from the cell wall, but the cell wall is still there. The cell wall is still the same shape it is pretty much for the other solutions. So the cell wall keeps the integrity of the cell. Okay, it's just the cell membrane is kind of shrinking away. So what happens there, the plasma membrane or cell membrane is shrinking away. That's called plasmolysis. Okay, so think plasma membrane, lysis, just think kind of like shrinking away. Okay, as the water is leaving the cell. Okay, but the cell itself, or the cell wall, is still there, maintaining like the shape. So the leaf itself, it might be drooping. If you forget to water a plant, the leaf is drooping, but it's not like completely shriveled up unless you really forget for a long time, and then it dies, right? So there's a difference between plant cells and animal cells when it comes to tonicity. So animal cells, hypotonic solution, they burst or lyse. Hypertonic, they shrivel or crenate. Plant cells, you have the turgor pressure that prevents the bursting, and then you have the plasmolysis that causes them to kind of break away from the cell wall, but the cell wall is still intact. Okay, so we're going to go into some review questions. So water moves via osmosis. So which one of these is the correct ending here? From an area with a high concentration of other solutes to a lower one. From an area with a low concentration of water to one of a higher concentration. Throughout the cytoplasm. Or from an area with a low concentration of solutes to an area with a higher one. So water moves via osmosis. So this is a tricky one. So think of water, the concentration of water moves from high concentration of water to low concentration of water. But here we know that this one's not right because this is the opposite. But these two are talking about solutes. So if we're moving from an area of high concentration of water to an area of low concentration of water, then let's relate that to solutes. So if we're moving from a high concentration of water, then that must be a low concentration of solutes. If we're moving from a high concentration of water, low concentration of solutes to a low concentration of water, it must be a higher concentration of solutes. So D is the correct answer. So that's a tricky one. Here's another question. On a hot summer day, you forgot to water your plants. Yeah, I do that quite often. They are now all wilted and drooping. What happened to the water inside the cells? A, the water moved into the plant cells, increasing the turgor pressure. B, the amount of water stayed the same inside the plant cells, causing no change in turgor pressure. Or C, the water moved out of the plant cells, causing plasmolysis. If you said C, I would agree with you. So plasmolysis, remember plasma means like plasma membrane, and then that's like the plasma membrane's kind of separating from the cell wall. It's kind of shrinking away from it. All right, so that completes part 2. What is passive transport? So by now you should be able to answer the following learning objectives. So you should be able to describe the processes of osmosis and diffusion, so both simple diffusion and facilitated diffusion. You should be able to define what isotonic, hypotonic, and hypertonic means in relation to solutions. You should be able to define turgor pressure and plasmolysis when it comes to plant cells, as well as describe the factors that affect how molecules move from one location to another location. So, think about like the rates, the concentration gradients, the size of the molecules, the solvent density, things like that can affect the rate, temperature. As well as identify the function of the cell wall. Okay. So the next video, we're going to get into part 3 of our building blocks of life, which gets into the chemistry part. So we're going to identify the biological molecules, so particularly the 4 macromolecules of life.